and a Washington Sea Grant Hirschman Fellow. And her talk is Exploring Spatial and Temporal Variations in Pigeon Guillemot Foraging Using Discarded Eggshells. It may have changed a little bit. Looks like it did. Anyway, here we go.
Thanks, Brendan. Well, thanks so much for having me, guys. I want to share Amanda's sentiments that I'm really excited to be here. Also, I want to thank Amanda for her presentation. She's a tough act to follow because she does really cool work, and she's been a really amazing collaborator with me and a great mentor to me as well. So a lot of thanks to Amanda for a lot of things. But today tonight I'll be sharing the results of what we called the Eggshell Project, as you can see in these pictures behind the pictures here on the screen. And I was able to tell you a little bit about this last year in person then, so missing seeing you all in person. But this year I actually have more to tell you since I completed the project, and I'm now working on submitting a paper with Francis called Identifying Dietary Shifts in Breeding Pigeon Guillemot. So let me know if you're able to see my slide progress here. Sometimes it's a little tricky. Ooh, okay.
Not yet.
Is that better?
Still
Looks
on
like,
the first slide to me.
am I frozen on the same slide?
There we go.
Okay, I might have a little bit of a lag, so yell at me if I'm going too fast. Just wanted to do just a quick intro about myself if I haven't met you before. But I collaborated with this group for my master's thesis for the School of Marine and Environmental Affairs at the University of Washington. And I worked with you all during the 2019 field season in Whidbey and in South Sound. I believe this is a photo Ann Mills took of me when I was out with her and Terrence. And now I graduated from my program, finished my thesis back in June, and I'm now a Washington Sea Grant Hirschman Fellow working to coordinate a different research group network called the Pacific Northwest Crab Research Group over in Port Townsend. But I am still a birder at heart, don't worry. So this project began with asking this question: where are Puget Sound pigeon guillemot deriving their energy and nutrients? Or in other words, what are they eating? And how might their diet vary depending on location and season? So I set out to answer this question using two types of information. The first one is one you're all very familiar with. It's the observations you all make of what prey adults deliver to chicks. And it's one of the best pieces of evidence we in Puget Sound have available to us right now for what pigeon guillemot are depending on food-wise, given that they may consume a lot of their prey underwater and spend some of their time in the non-breeding season farther offshore where they're harder to observe. When we see them, what we see them delivering to their chicks is one of the best indicators of diet that we have right now. And the second piece of information I wanted to use, oh gosh, sorry, my mouse is being a little funky. So the second type of information I used were stable isotopes, which are biogeochemical tracers of carbon and nitrogen that we can extract from an organism's tissues and use as indicators of their diet and the environment from which they feed. So this is because organisms are literally made up of what they eat. The carbon and nitrogen in our bodies comes from what we consume. So that saying, you are what you eat, is true. So each organism has a unique isotopic signature because different animals are going to be eating different things, different ratios of things. So given this, you can actually plot an organism's signature on a figure and basically build a food web of all these different organisms showing who is eating who. So we needed to get pigeon guillemot tissue somehow in order to determine their signature. And we wanted to do this in an easy, cost-effective, and non-disruptive way, which is how we settled on eggshells. So in 2019, you all and myself were successful in collecting 34 egg tissue samples from the base of burrows from 23 sites on Whidbey and in South Sound, which I have on this map pointed out here. And then I took them back to the environmental chemistry lab at the Northwest Fisheries Science Center, which is a part of NOAA, to process them. So here's a great photo of Francis training some volunteers at Mutiny Sands, I think. And so while these samples were being processed at the lab, I first took a look at the observation data. And what you can see is that if we compile all the observations made by volunteers of prey delivered, on average, gunnel is the type of prey most often delivered to burrows. And that is actually consistent throughout all of these locations. So I split locations up based off of the basins that colonies are adjacent to. So colonies in South Sound are adjacent to the South Basin. Colonies on the west side of Whidbey Island are adjacent to the Admiralty Basin. And then birds, colonies on the east side of Whidbey Island are adjacent to Whidbey Basin. Sorry, it's a little bit of a mouthful to say all of them. But the reason I split all these up by basin is because that's where we're assuming adults are catching these prey. So after looking at your guys' observation data, and this is from multiple years of observations, we got to the isotopes. And of course, lab analysis is nowhere near as photogenic as fieldwork. So this is pretty much the most exciting picture I have of the lab work, all of my samples right before they go through the mass spectrometer. But the first thing we did was look at the signatures of individual birds and see if there were any differences based off of the location. So each point in this plot is a single sample or an individual female pigeon guillemot. And they're female because we were collecting eggs, right? And the females are the ones laying the eggs. And it's showing their unique tissue carbon to nitrogen makeup. I say carbon to nitrogen because you can see that carbon and nitrogen are the axes of this graph. So the points are the ratio of those two isotopes. This slide might look familiar to folks who saw my presentation last year because I shared it last year. This was about where we were at this time last year. But it's still really cool because what we can take away from this figure is that birds breeding in these different locations have different diets. And that's why they're showing these different signatures. Again, so you can see the green triangles are individual birds who were foraging in South Sound, whereas Admiralty Inlet birds are the ones in the red, and you can see there's a significant difference between where those dots are located. So to progress farther, in order to start identifying what the pigeon guillemot are eating using isotopes, I needed to get the isotopic signatures of other organisms living in the marine system in order to construct a food web. And luckily for me, one of my mentors, Paul Chitterow at the Science Center, had been collecting fish and invertebrate samples for isotopic analysis for his own research. So by combining his data with mine and your guys' data, we could plot all of these organisms together using those distinct signatures, which now we've averaged. So we're going from single samples or single individual signatures to just an average, to show how they're relating to one another. So here you can see that photosynthesizing organisms are here at the bottom. So kelp and eelgrass. We have shrimp and invertebrates and fish here in the middle. And then we have the pigeon guillemot here at the top of this simplified food web. So once we'd gathered all of these data, we could then input them into a model that can tell us what the proportion of a pigeon guillemot's diet that these different prey species actually make up. And this is what the model spit out. that nearly 50% of pigeon guillemot diet is salmon. And we're assuming in this case that it's salmon smolts, since that would be, well, that would be the size that a pigeon guillemot could eat. And note that this part of the analysis was only performed for would-be birds, since that's where all of the prey isotopic samples were also taken from, and wanted to be consistent spatially. So, key takeaways. You might be thinking that I just gave you some conflicting results, that observations are saying gunnel, while isotopes are saying salmon. But these types of data actually provide us different seasonal information. Observations are made in the summer, while the isotopes only reflect the diet of female birds when they're gestating, which is a late spring diet. So you can see together, observation data and isotopic data provide information about pigeon guillemot diet during the 2 periods of the breeding season. This seasonal dietary shift is consistent with a pulse of spring salmonid smolts, and the spatial variation that we could see between birds at different locations that I showed earlier on may suggest differences in prey availability in the spring. However, your observations show that overall, adult pigeon guillemot prefer the same types of prey to deliver to their chicks, the gunnel. And this is consistent with literature that shows that adults that specialize in their prey delivery to chicks are more reproductively successful than those that generalize. However, gunnel are a low lipid, or you can think of a low fat, content fish, which is less preferable than a fish like a sand lance that is a higher lipid content fish. So I'm hypothesizing that it's likely that the reason pigeon guillemot, Puget Sound pigeon guillemots specialize in this less preferred prey, which are gunnel, is because that they're the prey that is most consistently available to them. So ultimately, what I believe this study shows is that, one, pairing these 2 different approaches, your observations with the isotopic analysis, these 2 different approaches to studying seabird diets allows us to see a more complete picture of how these birds are interacting with their environment. And the second thing that I think this study shows is that these birds do reflect, or one could say indicate, the variations in their nearshore environment, meaning that, at least in my opinion, they're very useful species to be watching in Puget Sound. So I went really fast because I wanted to make sure there were time for questions. But first I want to kind of echo what Amanda said, but thank you all so much for your contribution to this research. As you could tell, a lot of the data is coming from you all. So thank you again for, like Amanda said, your hard work and your contribution. I would love to take questions I know isotopes are kind of a tricky and sometimes confusing thing. So if I went too fast, I'd be happy to clarify. And then also, you know, I'm writing the paper now, and what I found is getting feedback from you all, your thoughts, your hypotheses, your observations, make me a stronger scientist and make the research stronger. So I would definitely appreciate if you want clarifications or feedback or anything like that. So thank you. And I can stop sharing my screen, but can go back if people would like to see any of those figures again.
Thank you so much, Emily. We can jump into questions, parents.
Great. Actually, before we get into the first set of questions, Emily, if you would mind putting in the chat your contact information for anyone who would like to get a hold of you. Just got a private message from someone who says that we actually here in Southtown have some information that would support your hypothesis about food fidelity, and you might be interested in reading that. So, okay, first question. I have observed pigeon guillemots on the water holding prey for a long time. Does that mean they're waiting for the opportune time to take prey to burrow, Or is that just how they eat for themselves?
Yeah, that's a really it's a really interesting observation, and I've also seen the same thing, that sometimes you just watch them sitting there with, you know, prey in their beak for a really, really long time. I know I've talked with Frances about this, and I think her hypothesis is that a lot of the feeding that the adults are doing is underwater, which is why it's kind of hard to know for sure what the adults are eating, and why I think the isotopes are really helpful in starting to get a sense for that. My sense, just because I've witnessed the same thing, is that it seems like they're just waiting for the right moment to take off and go to their burrows. They seem to be so, you know, so skittish and nervous about getting into the burrows. Eagles and just, you know, probably even wind, I don't know, is impacting them getting right in. So I would guess that it's them just kind of hesitating. But it's interesting to think about. So it's a cool observation and food for thought.
I think a lot of it might be eagles, at least in our area. They almost always, if we've seen that they're waited and waited, there's an eagle sitting in a tree. It might not be where I can see it, but the other people on our crew very often can see him because we're so close to the bluff, and the trees are pretty tall. So I don't always see him, but at the end of the time, when I've been watching this thing sitting out there for so long, then when I walk back and talk to our crew, the rest of our crew, they've seen the eagles. And the eagles seem to be getting to be more of a problem as far as keeping them from feeding.
Very interesting. Okay, so I got an isotope question here. Does the isotope for salmonids indicate which species of salmon is in the diet of the females?
Great question. No, it doesn't. Right, we have the data that I got from my mentor, who's the one who collected all those fish samples. They were collecting just a bunch of species. They were collecting the smolts, which are much harder to identify. So they kind of just collected a bunch and they've just combined them. So I think if you took, you know, if you had all 5 species of salmon and you knew exactly what they were, and you took those isotopic signatures, they would look ever so slightly different. But when you're looking at a salmon versus a gunnel, those are going to look really different, and the salmon are mostly going to all be the same. So for all intensive purposes, we kind of just lump salmon as one type of fish that they could be feeding on. But it would be really cool to start picking apart, you know, are they eating Chinook salmon, you know, or we hope not. No, so it'd be interesting to actually dig into that question more, but a really good one. And unfortunately, we're not that fine scale yet.
Could you get that kind of information from a hatchery?
Yeah, I mean, you probably could. Yeah, you could go and get, you know, you could go and get samples of exactly the type of salmon species you're interested in. What is challenging with the isotopes is that different organisms will have, you know, we can be like, that's generally a person, that's generally a bird, you know, we can see that, but then it does actually differentiate on an individual level, just because it's based off of what you're eating. So a hatchery fish and a wild fish might look a little different isotopically because they're feeding on different things. So it gets tricky for sure. But yeah, that's a really interesting idea. I love that idea.
Wouldn't the smolt isotopes come from which watershed they were coming down into the sound? So it wouldn't just be species specific, it would be watershed specific.
Absolutely. Yeah, you're absolutely right. And then it depends on how long these smolts are spending in different nearshore environments. So theoretically, a smolt in South Sound might have a slightly different signature than a smolt in Admiralty Inlet, just because the the ocean, those basins have different oceanographic chemistries. They have different habitats there. They're different ecosystems. So that's why isotopes can tell you a lot about the immediate system that an organism is living in. But it gets tricky when you have an organism like a salmon that is moving around a lot, right?
I just wanted to jump in real quick and say we're approaching 8 o'clock, and Emily is graciously said she'd stay on to answer other questions. But if for folks that do need to trickle out, thank you for being here. But we'll continue on for a little while longer. But we won't keep Emily too much longer.
No problem at all. It's COVID. I don't have other things going on.
All right, so do you anticipate that adult pigeon guillemots are eating the same prey types during the breeding season that they're feeding to their chicks?
Okay, so that's been the big question. I think that's something that Francis and I have talked a lot about, because like I said at the beginning, the issue is that we aren't really observing, well, you all and we all aren't really observing the adults physically eating prey. So we know what they deliver to their chicks. We don't really know exactly what they're eating. But like I kind of said earlier, what you all can see them delivering to their chicks is the best evidence for, what they're going after out there. And I think until some new evidence presents itself, I kind of hypothesize that if they're spending so much time foraging for gunnel and sculpin, that they're probably also eating them. So I'd actually be curious to hear other people's thoughts behind that. I think I've been a little bit working off of the assumption that during the breeding season, an adult's diet is probably quite similar to a chick's diet, just because of the energy it takes to forage. But I'd be actually curious if people disagree with me on that, because that's totally a hypothesis. We don't know for sure. We're having to make assumptions.
Okay, great. We got a couple more salmon questions here, and then we've got one that's specific to your study. So I want to actually address the study question first, since we've got a little bit limited time here. Is your sample size of 34 eggshells big enough to give you confidence in your conclusions?
That's a great question. So I think that sample size is large enough to kind of have these initial results, these kind of initial conclusions that we've made. What I'd love to see going forward, if this sort of research, this isotopic research, was continued, I think you'd want to have a much larger sample size, because that would be able to tell you a lot more about kind of more finer scale details. So you could get a better sense for, each colony having a pretty distinctive looking dietary difference. So I think for 34 samples is fine for making these kind of general observations and kind of like lumping all these species together. But for us to pick apart these really, really interesting questions about like the success of each colony and what each colony is feeding, what every individual is actually feeding on, you would need a lot more samples. So yeah, I think just there's a lot more exciting research to be done.
Very interesting. Well, I'm going to combine the last two salmon questions into a two-parter. So one person is trying to understand, she thought that salmonids were benthic, like the fish that the pigeon guillemots prefer. She's under the impression that they are benthic fish, or like benthic fish. And then the other part of the question is, do you have a way to differentiate if the salmon component of the diet, if that's by region or if that's by just kind of a general grouping?
So the question of whether I know if they're eating salmon in South Sound as well as Whidbey, is that what you mean?
Yeah, yeah.
Okay. So to answer that one first, so we can only say that Whidbey Island birds are eating the salmonids. And that's because the data that I used in the model, this like the fish isotopes, those were all collected by Whidbey Island. And kind of like I was alluding to before, that that organisms in different regions, even if it's like salmon in South Sound and salmon in Whidbey, might look different isotopically. So we don't want to use a fish isotope from a different location and apply it to Whidbey. So that's why we can't say if the South Sound birds are eating salmon. However, the reason why I wanted to leave in that slide showing that Whidbey birds and South Sound birds have pretty different isotopic signatures is because what's interesting is that we know that they're all delivering gunnel to their chicks, no matter where they are during the breeding season. But we can see from the isotopes that they are eating different things in the spring. So my hypothesis there is that South Sound birds are feeding on fewer salmon than Whidbey birds are, or they found some other pulse of fish that they're preferentially feeding on in South Sound, and that would make sense, right? That these are two really different oceanographic basins that have different freshwater inputs going on, and probably very different salmon abundances in those places. And then to answer the other question that you posed, Terrence, so salmon are I don't believe are considered a benthic fish. Gunnel and sculpins are both benthic fish, and that's and I think you're totally right. Whoever wrote this question, that generally pigeon guillemot seem to prefer benthic fish. But I think more than preferring benthic fish, pigeon guillemot are opportunistic foragers who are really taking advantage of what's in the system. So that's why kind of my kind of last takeaway of the presentation is that we're seeing them so consistently deliver gunnel and sculpin because that's a prey that's consistently available to them. Whereas we're seeing something like salmon pop up in the spring because there's a lot of salmon in the environment in the springtime. And so they're going to take advantage of that. So I think that what we can see from this is that these are really, really well adapted birds to their environment. They are making the most of every opportunity that's out there, and eating whatever they can find and find often.
So a follow-up question to that from myself. Do you then consider pigeon guillemots to be a species that's kind of occupying a specific ecological niche in specializing in these benthic species, as opposed to, making the trade-off of pursuing higher lipid content fish like salmonids or other forage fish species that may or may not be present in the areas where they're breeding?
I think that's a really interesting question. That's kind of where, we have to be looking at the literature and these other studies that have been done in Alaska or in California to kind of start getting a sense for this life history. And it's interesting because it's really all over the place, that studies that are done in different parts of the West Coast show these birds, kind of having different diets. And I think what that shows is just, like I kind of said before, that they're just really opportunistic, and if you're going to have something like a high lipid content fish, like a sand lance or a salmon that's in your area, they're gonna go after it, and that's gonna be their preferred prey. But I think what it might show is that there actually aren't as many high lipid, like preferred prey items in Puget Sound as might be desirable for these birds. And so it makes me think a little bit about what's funny as a side note is that this project I went into being like, this is a bird project. I'm like an ornithologist. And it's actually, much more about fish than about birds, to be honest. The birds are a way to learn more about the fish, it seems. But so that was, I think what's interesting is, to me, what this study shows is that Puget Sound might, there might be issues with the fish populations, that these birds aren't feeding on what they'd probably like to be feeding on, but they kind of eating what they can get. So I don't know, it'd be interesting to have a natural fish person in here to maybe, talk about that more, but that's a little bit what this study has made me think about.
Well, great, thank you. That wraps up all of our questions. You'll see some thank yous in the chat there for you, Emily and Amanda. And I'll turn this back over to Brendan.
All right. Well, I don't have a whole lot to say besides a bunch of thank yous to everyone. Everyone for being here, everyone for doing your surveys in this kind of strange year that we've had.